Hello, good morning, good afternoon, good evening, wherever you are. Welcome to another session to this gene therapy course, DNA rejuvenation technology. And in this session, the ninth one, we're going to see the adeno-associated virus plasmid design that you have here. We're going to see how to design this kind of plasmids that carry information for amplifying adeno-associated virus plasmids in E. coli, in bacteria. You see here, this plasmid consists of two parts. This first part consists of information necessary to amplify the plasmid in bacteria. And the second part that you see here, this information is necessary to amplify the genes of interest in mammalian cells, specifically in HEC293. This information carries the necessary instructions to amplify these genes in mammalian cells and to express these genes in human cells or another kind of cells. What we're going to do is understanding each of the parts. You see here the promoter, the resistant gene, the ORI, and so on. And once we understand the different parts, what we are going to do is copy pasting one after another. And we are going to use text editors. As you can see here, we can use a text editor that is specific for DNA editing, as this one that is called serial cloner. You see here the sequences, one after another. They are highlighted in colors. For example, this sequence corresponds to this one, this sequence corresponds to this one, and the same here. Instead of using a specific text editor, you can use just Word, like in this picture. So let's go for it. Let's dive into this information. Let's learn a little bit. Well, here we are. Let's go for it. Okay. As I have told you, these plasmids consist of two parts. Let's go for the first one. The first one is being highlighted in blue font. You see here the ORI, the promoter, the resistant gene. Okay, this information is necessary to amplify the plasmid in bacteria cells. You see here we have produced many copies of this plasmid in bacteria cell. We introduce the DNA inside bacteria cells through a transformation process, and we amplify the DNA. Okay, so let's have a look here. The most important part is the ORI sequence. The ORI sequence is responsible for the duplication of the DNA. The bacterial cell recognizes the ORI region, the ORI sequence, and start duplicating the DNA thanks to DNA polymerases. What we have also here is a promoter and a resistance genes. The promoter is preceding the resistance gene. The promoter is necessary to express the resistance gene. The resistance gene is a gene that codes a protein that make the bacteria resist to antibiotic. It may be ampicillin, tetracycline, kanamycin, or whatever. This is necessary for this resistant gene is necessary for selecting a bacteria that has the plasmid. So only the bacteria carrying the plasmid will grow in the media. So at the end of the process, we will have a lot of bacteria with a lot of plasmids. And that's all, my friends. That's all that you need to know here. We will only require three sequences regarding this part of the plasmid. Let's go for the second part of plasmid. You see here the one that is highlighted in orange. Okay, this part of the plasmid is responsible for amplifying this region of the DNA, just the gene of interest genes. Okay, that part of the sequence is responsible for amplifying, producing many copies of this inside mammalian cells. You see here, there are many copies. And the responsible for that are the ITR sequences. You see here 1 and 2, one in the 5' side and the other one in the 3' side. They escort the genes of interest. It's kind of a label. So whenever the cell recognizes these sequences, the ITR on the 5' and the ITR in the 3', the cell will start the cell, thanks to another two plasmids that are here, that you know, the red cap plasmid and the helper plasmid. It will start producing many copies of the OS genes. And also these sequences, the ITRs, are responsible for packing, encapsulating these genes inside the adeno-associated virus. So the HEC293 cells, the mammalian cells, will recognize whatever is inside the ITRs. It will start amplifying these genes, and afterwards they will start encapsulating the genes inside the adeno-associated virus. Okay, so this will be the treatment of the adeno-associated virus with this information that is in between the ITRs. And then the adeno-associated virus with the genes are delivered to the human body. The virus get to the cells, go to the nucleus, dissolve its capsid, and deliver the genes inside the nucleus. Once the genes are inside the nucleus, they will produce proteins, and these proteins will be the treatment. Okay, this is the treatment that produces the magic. Okay, regarding this side, the part that is necessary for amplifying the plasmid, we know everything that we need to know. There is only required three. Uh, it's only required these three pieces of information: the promoter, a resistant genes, and the RI. Regarding this part, we have to learn other components that are inside. Okay, so let's go for it. Let's go to dive into this information further. Let's go for it. Now you understand this part of the plasmid. It consists of three parts: the promoters, the resistant genes, and the RI. I will leave the sequences in the description below. And now we know the ITR sequences and also the genes of interest. So in here, what we are going to do is breaking down the gene of interest sequence. It consists of many other sequences. Okay, so let's go for it. First, what do we see? We see the restriction site 1 and the restriction site 2. We have selected within the multi-cloning sites that are here, we have selected the two restriction sites to clone this information inside the plasmid. What information have we cloned? The following one. Okay, but the first thing that we require is two ITR sequences. The ITR sequences are the inverted terminal repetitions. That stands for inverted terminal repetitions. And we need one in the 5' and another one in the 3' side. So here we have the 5' one and the 3' one. These sequences, as I have told you, label what is inside, what is in between them. So this information is necessary for amplifying whatever is in between them, and then to encapsulate whatever is inside them inside the adeno-associated virus. Okay. So there we are. We will require these two sequences that will be escorting, that will be labeling this part of the DNA. And then what we need is just genes. For whoever is reading this or listening to this or watching this, most likely you know how a gene is. Normally, you have some information that is necessary for transcription and other information that is necessary for translation, right? And you normally have one gene. In this occasion, you have two cassettes of genes. For example, you have here the first cassette of gene, And here the second cassette of gene. These are the genes that are going to express in mammalian. These ones that are encircled. And these genes are necessary for controlling the production of the second one. So the first gene control the production of the second one, and the second one is the treatment in this occasion. Okay, so let's go for it. Let's see first the first gene and the information involved in the transcription of this gene. You have first a CMV promoter, acetomegalovírus promoter. It's a constitutive promoter. Constitutive promoters means that it's constantly transcribing, promoting the transcription. Okay. Then you have a poly A. So the promoter, what it's going to do is initiating the favoring the initiation of the transcription of this fragment. And you see here the poly A. The poly A is the sequence that means that it has to stop transcribing. It will add a tail of adenines at the end of the mRNA. So the promoter will start producing the mRNA in the starting site, and the poly A will be the stop signal. It will produce a mRNA like this. Okay, this is the information necessary for the transcription, the CMV and the poly A. I will leave the sequences below. Then for translation, what do you need? You need a COSAT site, a initiation codon, and the COSAT will be information necessary for locating well the ribosomes in the correct position, so that they start translating in the correct position. And then you have the information necessary for this gene. This gene codes a protein that binds the oxycycline, and once this protein senses the presence of the oxycycline, we administer the oxycycline, for example, to a patient. This deoxycycline binds the protein that is called transactivator protein RTTA4, and it binds to the second promoter. By binding to the second promoter, induce the transcription in the second cassette of genes. Okay, so that is it. The first gene consists of the CMV promoter, the poly A signal. This is for transcription, and then you have inside information necessary for translation. This is the Cossack sequence, and then the transcription activator genes. Okay, very easy. It will produce this protein. This protein will bind the oxycycline, and once it is bound to the oxycycline, will be able to activate the second promoter. Let's see the second cassette of genes. In this cassette of genes, will produce a polycistronic mRNA. That is to say, it will carry information necessary for three genes. In this occasion, that we are studying the reversion of the DNA age. So in order to do that, we need to deliver three proteins to the cell that are called OSK. Altogether are called OSK, and it stands for OCT4, SOX2, and KLF4. Let's go to see how it works. It's similar. Okay, first thing that you need is a promoter. The promoter will initiate the transcription up to the poly A signal. The poly A, remember, is the signal that means the end of the transcription. Okay, once the RNA polymerase detects the poly A, will add a tail of adenines, and it will stop producing the mRNA, as you can see here. You see the mRNA, and the mRNA will go out of the nucleus, and it will be translated. Okay, so the first thing that you need is a promoter and a poly A. And in this occasion, you have a regulatory element preceding the promoter. That means that this promoter is going to be regulated by this protein that I told you, the RTTA4. Okay, so in this cassette of genes, you need the regulatory element, a promoter, and poly A. This is necessary for transcription, and then you have information necessary for translation. That is first the cosas sequence and the cassette, second cassette of genes. You see here, this is the first gene, the OCT4, the SOX2, and the KLF4. And in between, you have sequences that are called P2A. P2A. This is necessary for producing proteins just using one mRNA. The ribosome will bind here, around here, and will start translating. Thanks to the Cossack sequence, we'll start translating the OCT4, and once it is detected, P2A will produce a gap, and will carry on translating the SOX2. In here, we'll detect the P2A again. It will produce a gap, and will carry on producing the KLF4 until it detects the stop codon. Once it detects the stop codon, the production will be finished. As you can see here, you have the 3 proteins produced, right? So that is all that you need to see. Let's remember a little bit it, okay? Well, and once we understand the different parts of the plasmid, the only thing that you have to do is copy pasting one part after another, one sequence after another, and you can use, for example, a specific DNA, specific text editors for DNA, for example, Serial Cloner or a Snap Gene or another software, or you can use just simply Word. I have to say, if you are going to go for Word, the Microsoft Word, try to use a lot of colors, identifying every single part. Otherwise, it gets messy because it's a lot of information, it's a lot of text. You see here, right? Every single part of the plasmid has a different sequence, and I have highlighted the different sequences in different colors. For example, this one that is orange corresponds to this part that is orange. The ITRs, you see here, this is the sequence for the ITRs. You see here, for example, here is exactly the same. Using this other editor is exactly the same. The orange part, in fact, correspond to this, so it's bigger. For example, you see here the promoter represented here, the resistance gene represented here, the ORI represented here, and here you have the sequence for the ORI, the sequence for the OS genes. Okay, important things to mention. Let's go for it. You see here, let's remember a little bit the parts. Okay, the restriction site 1 and 2, flanking, escorting the whole sequence. Then you have the ITR sequence, the 5' and the 3'. These are, remember, the labels to amplify this region and to encapsulate in the adeno-associated virus. Okay, and then you have the 2 cassette of genes, this first gene and the second cassette of gene. And inside a gene, what you have is, for transcription, you have a promoter and a poly A signal. This is for transcription, for transcribing the first gene. And then inside the gene, what you have is information necessary for translation. That is the Cossack sequence necessary for locating the ribosome in the correct place, and then the information of the genes with a start codon and stop codon at the beginning and at the end. Okay, the protein that will be produced here will be necessary to regulate the second cassette of genes. In the second cassette of genes is similar, but it extends a little bit this information. You have first a promoter and then a poly A signal for transcription. The promoter includes another element that is a regulatory element. This regulatory element requires the presence of another element to start producing the transcription. Okay, this is what it needs is the transactivator bound to the oxycycline that we give to the patient. So it starts producing the mRNA here, right? And it stops here after the poly. So afterwards we have the mRNA. The mRNA is called 3 proteins. The ribosome will start producing, once the ribosome sees the COSA sequence and the start codon in the COSA sequence, it will start producing these proteins. We'll start producing OCT4, we'll produce a gap here to have it separately, then it will carry on producing the SOX2, it will identify the sequence, it will produce a gap, and then it will start producing the KLF4 until it sees the stop codon and it will stop translating. At the end of the process, you will have these 3 proteins, right? So, and what happened in between these sequences? These sequences are specific, right? I will leave the sequences below in the description. But these sequences are random sequences between 10 and 20 base pairs long. Okay, I can leave random sequences that you can use. Some of them have to be in frame. That means that they have to respect, they have to be multiples of 3. Why? Because of the codons, right? In between certain elements, you have to be always in frame. And which are these elements? Okay, so you see here in between the Cossack sequence and the transactivator gene, you have to be in frame. It's important to be in frame here in Cossack, in between Cossack and the transactivator genes, and also here in between Cossack and the rest of the genes. These 3 genes and the P2A sequences have to be in frame. Cossack has to be in frame with OCT4, OCT4 with P2A, P2A with SOX2, SOX2 with P2A, P2A with KLA4, and that's it. Okay, at the end of the process you will have to check with a translator that every 3 nucleotides that you have multiples of these sequences are multiples of 3 nucleotides, and the distances between these elements are multiples of 3 nucleotides. Okay, so that is what you require. Now you know understand the different parts of the sequence, and what you have to do now is copy pasting. I'm going to leave the sequences below, so you can play with them separately, and also going to leave the whole sequence of the plasmid below. So that was all for today, my friends. I will extend a little bit this information in following videos. I will show you how to edit and doing these things. Okay, but for today is more than enough.